Yeah, that's good. That's great. Yeah, good, good, good. So that the lights get up. That's good. Okay, and the mics are already on, so that's good. Okay, good. I'm gonna go back to my call.
Yep.
Just gonna check real quick. About 25 people. 26.
Yeah, it's decent. I'm impressed. I'm a good number on popping in online too. They should start coming in. Would you prefer a lavalier microphone so that the Zoom folks can hear you?
What do you think is better? This one? Doesn't matter, I'll do whatever you
Oh,
tell me.
I think this is probably the easiest. for
Okay.
you. That way you can walk around if you want. There's a laser pointer if you want. I don't have a slide.
Okay, no problem.
And I do have just a few slides that, the typical introductory slides that we do. Okay. Then once this is done, we'll close those out. We get people coming in.
Great, thank you.
Anyway, let's go ahead and get started. Thank you for coming to our meeting, and I know we'll have a few people drifting in. So I'm Jason Heindel, and it's my pleasure to welcome you and to welcome Cesar to give this talk today. A few introductory slides for you. Hopefully everyone on Zoom can hear us. If not, we'll fix that offline in a little bit. So before I introduce our speaker, just some branch items. First off, of course, we have to encourage you to join the branch and to join ASM National. Why might you do that? Here are some reasons: to expand your network, participate in the microbiology community, to enjoy some awesome science, of course, and to participate as a scientist and read about science. The fees are reasonable. The fees just went up, so it is on a calendar year basis. So those are the current fees. If you want to join, more info is mpaasm.org. or at asm.org for the national fees. Notably, to join the branch, it's free for students. You do have to join national, or you have to log in through national. So you don't have to join the pay. So affordable for students, not so bad for non-students otherwise. Here is a list of our upcoming events. We have a monthly meeting. We have several upcoming. The 817th meeting in February is Andrew Urachko, who is currently ASM Distinguished Lecturer. He's at LSU Health in Shreveport, works at HCMV, so he'll be coming to give our talk then. Our own Robert Potter from CHOP will be in March. In April, we have Carrie Ann Byrne from Pattern Biosciences. And in May, the student chapter is organizing a meeting. TBD on who will be the speaker there. So stay tuned for that, and please consider coming to the meeting. We also are having season 4 elections. Branch officer elections will be happening. You will receive information from that if you're a member of the branch via email. We have president-elect, secretary, and treasurer are in the running and available for election. A nominating committee will be coming up with a slate of candidates. However, if you yourself would like to, or know someone you think should be nominated, you can petition for them to be included. More info for that is on the branch website as well. Get that to you. And also, we are actively looking for applications or nominations for the ASM Peggy Cotter Travel Award for early career branch members. This is a great opportunity to get some funding to go to the national meeting, ASM Micro, which is in Los Angeles this year. We typically have 2 or 3 folks from the branch receive this award and travel to the meeting. So do think about it. If you know someone, if you are that someone, please apply. There's more information on how to apply on the website as well. We'll be accepting applications through this February 21st. So please pay attention to that. We may get an email about that if we can get our email system this second. All right. So now, oh, and if you want parking after this, I do have parking passes for those of you who need the park. Let me know. All right, so it's my pleasure to welcome our speakers that are Cesar de la Fuente from Penn. He's currently a presidential associate professor with too many affiliations for me to name here. You can look them up yourself. And I do have just some highlights of his career. So he took his bachelor's and master's degrees at University of León in biotechnology. Following that, he did his PhD at University of British Columbia in the laboratory of Bob Hancock. And then he small postdoctoral stint there, and then moved to MIT for his postdoctoral fellowship with Tim Wu. Since coming to Penn, he has received numerous awards. Basically, if there's a number under number award, 30 under 30, top 40 under 40 award, he's been receiving those kinds of awards on a weekly basis right now. A couple ones to note: the Fleming Prize from 2024 was one of the recent awards he received. The ASM Award for Early Career Basic Research in 2024, he just received. He was a distinguished lecturer in 2021 for ASM, won the Langer Prize for Innovation and Entrepreneurial Excellence, with the inaugural winner of that prize. Currently has strong funding, is a MIRA investigator, so an R35 H, has good funding from the Defense Threat Reduction Agency, and is a Brain and Behavior Research Foundation investigator. Hundreds of publications in journals that you have read or will be reading soon. Aside from the information he's going to tell us today, we have other stories in the lab, including molecular de-extinction, reprogramming BEMS for antibiotic design, and low-cost or rapid diagnostic tools for infectious diseases. So if you're interested in those, check out YouTube or his website. But today, he's here to talk to us about AI for antibiotic discovery. So please join me in welcoming Cesar.
Great. Can everybody hear me okay with this? Yeah. Well, thank you very much for the introduction and for the invitation, really, to speak here. It's it's really a pleasure to be here. So thanks. So I thought I would tell you about the work that we've been doing, applying AI and computational methods for antibiotic discovery. This is my lab, and this is something that I've been dreaming for a long time, sort of combining the power of machines with biology to be able to accelerate discoveries in biology and in medicine. And I've always thought, I remember when I was, even in my teens, thinking that biology was just this amazing thing that could serve someday as a general purpose technology, right? If we truly understood biology from first principles, we would be able to extract some of that understanding to try to develop new therapeutics, new medicines, new tools to target the biggest problems that we face today. And I remember then in my early 20s when I was doing my undergrad, thinking, what from biology, what could we extract from biology? Is there some sort of solution in biology that could be used to target the biggest problems in our society, right? And actually, I learned that there's a minimal functional molecule called a peptide in biology. This is, proteins and peptides are the nanomachines that do everything in life. They help us digest our food. They help me talk right now. They catalyze every reaction in our body. They influence the way we feel. They fight off infections. And peptides are just basically miniature versions of proteins, but they're actually also functional molecules. So they can do all of these things. And so suddenly in my early 20s, I had this realization that, if we could really program these small proteins called peptides, we perhaps would be able to tailor them to tackle some of the biggest problems. And throughout my whole career, I've devoted my time to antibiotics. And that's because around that same time, when I was doing my undergrad, I did an analysis on the biggest problems that affect the most people. And which of those problems were there was less money invested in trying to to solve those problems. And antibiotic resistance came up on top. It's actually the most underinvested area that affects the most people in the world. And so I thought, why not try to do something about it? Antimicrobial resistant infections, currently they're associated with about 5 million deaths per year in the world. The projection is by 2050, that number is going to double to 10 million deaths per year in the world. It's going to surpass even cancer as a cause of death in our society. So it's a huge problem, right? Those 10 million deaths in the projection, if you do a quick calculation, they correspond to about 1 death every 3 seconds. So really, I think is one of the greatest existential threats to our society, really, truly. And there's not a lot of money being invested. And so one of the goals of my lab is to try to think outside of the box to see if how we can come up with new ways of discovering antibiotics. And we, like I mentioned, we focus on peptides because they're nature's simplest nanomachines. And they're a gateway to understanding biology's complexity. If we understand how peptides work, that will increase our understanding of how proteins work and different pathways within the cell and different mechanisms within the cell. They also have an astronomical sequence space, so they're ideally suited for AI, right? Their sequence space is basically infinite. And so it would be unfeasible or impossible for humans to sample through it experimentally. So it's a perfect case scenario for AI research. Peptides are small and therefore also experimentally tractable and scalable. We have in my lab, we have robots that actually stitch the different amino acids together to make the peptide molecules, and we can do this in a very high throughput way. Peptides they're also very tunable molecules. So by simply changing your optimization function on your algorithm, you can tailor them to do different things. A lot of what I'll talk about today is to turn them to become antibiotics, but you can also change the optimization function to create anti-cancer molecules or immunomodulatory agents and things like that. And we're increasingly working on all those areas as well. And finally, just more conceptually, out of all of life's biomolecules, we have DNA, RNA, and proteins and peptides. The latter, they're the only ones that are still not programmable, right? DNA, we can read and write DNA pretty cheaply today. RNA, we can now make therapeutics and vaccines out of RNA. But amino acid-based molecules, proteins and peptides, we're still not quite there in our ability to program them. So I think it's an exciting new frontier in the years to come. Okay, so when I got recruited at Penn from MIT, when I started, I asked this fundamental question, which is, can we use AI to accelerate the process of discovering new antibiotics? And just to give you a sense of how typically people discover antibiotics, the traditional methods, it's an extremely physical process where people go around nature and you try to take soil and water samples and you try to purify active compounds that might be present in all of this complex organic matter. But as you can imagine, this is a process that extremely relies on trial and error experimentation, oftentimes does not yield any good candidates. So instead of doing that, we asked ourselves, why don't we take advantage of the decades worth of biological data that we have at our disposal in the form of genomes, metagenomes, and proteomes that have been sequenced over the last couple of decades? And why don't we develop algorithms to systematically and digitally sample all of those data to try to find new potential molecules that might be hidden or encoded within those data? So the analogy is kind of like our algorithms operate like a magnifying glass that enables us to look beyond what you typically see in data to try to find new molecules. And this is sort of how it operates. We can sample through, in this case, proteins with different proteins, and the algorithm sorts through them sort of like a barcode and is able to identify regions within that barcode that might be potential new therapeutics, in this case, antibiotics. Okay, so this was the conceptual framework that we came up with, just thinking of biology as a bunch of code that we can search through with the right algorithms, and then we can find new things that might be helpful. The other motivation for using AI was that it's very slow to identify preclinical candidates. It can take up to 6 years So that's more than the typical PhD program to just find a couple of potentially interesting candidates. And it's a super expensive process, right? Over $2 billion. So we thought with AI, we could dramatically accelerate all of this, right? That was the premise. So I'm going to tell you what we did chronologically, right? So you get a sense of also how we did the science and how we thought about the next steps along this journey. We started by taking inspiration from image and speech recognition algorithms. We thought, what if we develop similar algorithms, but instead of recognizing faces or sounds, we recognize molecular patterns that are present in biology, in a biological system, right? That could constitute a potential antibiotic, right? So we decided to develop pattern recognition algorithms to mine individual proteins to see if we could find fragments hidden within the protein that could become a new antibiotic. So a protein, we can display it in 3 dimensions, but we can also display it in 2 dimensions with the amino acid code here in the y-axis. And our algorithms, essentially what they do is they run through this code and they identify regions that are predicted to be antimicrobial. And these are given by these different colors here. It's only theoretical work, right? Because it's algorithmically driven, but it's quite powerful, even theoretically, because it allows you to explore individual proteins and identify regions within a protein that might be a potential antibiotic, for example, this region in yellow here. And then once we've done that, we can, using synthetic biology tools, we can extract that fragment from the protein and we can try to develop it into some new therapeutic, right, in the lab. Okay, so we did this for a number of projects. We were very happy mining individual proteins, but soon thereafter, we realized that we could scale up from mining individual proteins, with advances in compute power and algorithmic power. We came up with this idea of like trying to scale up this process. And that's what took us to embark on this journey of trying to explore the human proteome for the first time as a source of new antibiotics. And the human proteome are all the proteins encoded in our genome. So this was a huge scaling problem for us, right? We're going from 1 protein at a time to suddenly 42,000. We went beyond the 20,000 gene-encoded proteins. We took into account isoforms. So the final number was 42,000. So, okay, how are we going to do this from 1 to 42,000? So we ran a number of calculations. We thought this exploration driven by this algorithm would take, multiple days. But to our surprise, it was quite remarkable. After just 1 hour, the algorithm completed the exploration. We had overestimated how computationally inexpensive our algorithm actually was. And since people had not done this before, we didn't know where, our calculation was actually wrong at how long it would take. So in just 1 hour, this algorithm was capable of sampling all the genetic code, right, that makes us who we are. And it was able to identify thousands of new antibiotics that we call encrypted peptides that were encrypted in proteins from our body, including proteins that play a role in the nervous system, the cardiovascular system, digestive system, and immune system as well, that were completely previously undescribed. So with a simple algorithm here, we're uncovering a whole new world of things in our own body, right? And the final thing is that the hit rate was 62.5%, meaning out of the molecules that the computer predicted that would be active against pathogens, when we synthesized them and we did experiments, a little bit more over 62% were actually active, which is not amazing, but it's not bad, right? Just so you can keep a sense of the ground truth sort of experimental hit rate that we get. Okay, so then out of those thousands of new molecules that we discovered, we synthesized dozens of them, and then that was a hit rate that we got in experiments. And then we tested them in preclinical mouse models. This is one of them. This is a skin abscess infection model. It's sort of like a biofilm-like infection on the back of the mice. And just to give you a sense, these are 2 different experiments. The mice are infected with 2 different pathogens, Pseudomonas and Evomani. And in gray, we have the untreated control group of mice. And here we have treatment with 1 peptide, treatment with another. And then in purple, we have a cocktail of these 2 together. So you can see a significant reduction in infection in all these scenarios, right? And this was interesting because these are compounds encoded in our genome and in our proteome. So therefore, they possess intrinsic beneficial properties like, most likely lack of toxicity and things like that. Okay, then we found that out of the thousands of new molecules that we identified, some of them had played roles physiologically. These are 4 examples. We can see that they're produced at different levels from low to medium to high and in different parts of the body. The hypothesis that we are currently working on is that we think all these encrypted peptides, they play a role in host immunity. So when we have an invading pathogen entering our body, we think a lot of these get produced as the first line of defense to protect us from pathogens. And we think that there's a lot of what we call peptide-based immunity that was previously under-recognized, and how much a lot of these peptide molecules might be playing a huge role in the immune system. So this work really, it sparked a lot of new ideas in my lab and a lot of new hypotheses, one of which was the fact that we're finding suddenly thousands of new molecules in our proteome most likely means that there might be widespread, right? They might not just be there, but so we came up with this idea that we might be able to find similar compounds all throughout evolution and across the tree of life. And since we had focused initially this work on the human proteome, we decided to look at our closest ancestors for similar compounds. Our closest ancestors are Neanderthals and Denisovans. And in the process of thinking about all of this, we came up with a new framework for identifying molecules that we call molecular de-extinction, whose ultimate goal is to discover molecules throughout evolution and then synthesize them in the lab using chemistry, and then learning about how different mutations that occur over time influence their biological function. And because we focus on peptides, which are functional molecules, we can for the first time do that, right? The traditional paradigm is looking at DNA, which is a wonderful molecule, but it only tells you it's an informational molecule. It doesn't have a function like peptides or proteins, right? So now we can look throughout history and we can compare with present day biology, and we can see through phylogenetic trees how different molecules evolved and how that evolution and those trajectories influence their biological function, which can be antibiotic activity, but it can also be immunomodulatory activity, ability to target cancer cells, ability to interfere with neural pathways, and so on. So this is a new framework for thinking about all of this. Going back to ancient humans, to try to discover antibiotics there, we had to develop a machine learning model that we call PanCleave. I'm just going to tell you at a high level how this operates. It takes all the proteins from Neanderthals, let's say, it chews them up in little fragments, and then we run 2 filters, one of which is a machine learning filter, the other one which is a human expert filter, each of which predicts whether those fragments will be antimicrobial or not. And I highlight this because every critical decision in my lab takes into account the recommendation made by the machine, but also the recommendation made by the human scientist. And I think this is quite sort of predictive of how I think science is going to evolve. I think it's going to be this tight collaboration between machine intelligence and human sort of ingenuity or ability to contribute in the loop, right? So this machine learning pipeline, it enabled us to discover the first therapeutic molecules in extinct organisms. Here we have some examples in the case of encoded in the Denisovan proteins and in Neanderthal proteins. We use the modern human proteome as a control in everything we did here. Once again, this is only theoretical work. It's all predictions made by this AI model. But then in my lab, we make a lot of these using chemistry, and we do all the ground truth experimental validation. So we saw that many of them had activity in vitro against gram-negative and gram-positive pathogens. And this is the in vivo work in 2 different preclinical mouse models, including a skin abscess model here on the top, which is the same one that I described earlier. And here we have a deep thigh infection model, which is typically the one that the FDA here wants to see for exciting preclinical candidates. And Neanderthaling was the best candidate at reducing infections. We also had to come up with new names for all these compounds. So the nomenclature that we established was that we name our new molecules based on the organism that produces them. So Neanderthalin comes from Neanderthals and so on, right? So this was really exciting. It was the first application of molecular de-extinction as a practical framework that would allow you to discover new interesting molecules. And it gave us perhaps the courage to ask a more ambitious question, which was, why not just sample every single extinct organism known to science? Why not just sample all of ancient biology to see if we could find new antibiotics there? But once again, once we started thinking about this more practically, this imposed another scaling challenge. We're going from 3 organisms here to suddenly hundreds of them. So we had to develop a more powerful AI model. So we came up with Apex, which we developed and we trained from scratch in my lab. This is the architecture. As you can see here, it uses recurrent neural nets plus attention mechanisms, underpinning a lot of the chatbots that we use today, like ChatGPT and so on. The really cool thing about Apex is that all you give it is an amino acid sequence. And from that amino acid sequence, it's capable of predicting its antibiotic activity. So it's a sequence to function prediction model. It doesn't take into account structure. It obviates that step. So arguably, it's an even harder problem, right? You're giving it an amino acid sequence, and it's telling you what that particular molecule is doing out in the world. In this case, killing bacteria. But you could apply a similar concept to giving it an amino acid sequence and the model telling you, can this kill cancer cells, for example? So sequence to function prediction directly. Very difficult problem, as you can imagine. That was the whole premise. Can we design function directly from sequence? And Apex was capable of doing that. Training Apex. This is I like to tell the historical context of everything that we do in science because it's interesting, I think. Training Apex was non-trivial. So when I came to Penn in 2019, I knew that for AI to be successful in biology and in the field of antibiotics and microbiology, we needed really good data sets. So I decided to design a standardized data set in my lab. And so I convinced my team to run all these experiments where we had different molecules and the respective antimicrobial scores, all through experiments against many different pathogens. And in my initial calculations, I thought this would take about 2.5 years to collect enough data to be able to train Apex. So after 1 year of starting to collect data, we tried to train Apex. It failed. The metrics were just not good. After 1.5 years, we tried again. It failed. 2 years, again, it failed. And I was starting to get nervous at that point. 2.5 years, it failed again. We just didn't have enough data. Again, I had run these calculations, but people had not really done this before. So I started to worry at this point because I said, maybe what if I'm off several orders of magnitude? Maybe this is not a several years long project. Maybe this is a decades long project, in which case it's unfeasible in the context of a academic lab, a PhD, and I don't want to be 80 by the time that this works. And then 3 years, it failed again. And after 3.5 years, success. So it took us 3.5 years of collecting experimental data and into a standardized data set to then feed that to Apex for Apex to become the world's best AI model at predicting antibiotic activity from a sequence. And it was a huge relief, right? Because otherwise my team would have killed me. I would been telling them for 3.5 years, this is going to work. And then thankfully it did in the end. And as you'll see, this opened up a whole new era for this field. Apex opened a window into the past. It enabled us to sample many organisms throughout evolution. Here are some examples. And here's a diagram showing how we were able to sample all the way back to the Holocene and the Pleistocene. Here are representative organisms where we found the top antibiotic candidates. It includes penguins that were extinct in the 50s, zebras that were extinct in the 1800s, magnolia trees that disappeared over evolution. It includes ancient elephants, woolly mammoths, giant sloths, and many, many other creatures from the past. So we were able to essentially sample all of ancient biology to try to find new molecules of interest. Then we synthesized a lot of these using chemistry, a lot of these molecules that we found. This is just to give you a sense of the magnitude of the exploration driven by Apex. And we have both predicted and active. These are experimentally validated. Hundreds of organisms, virtually all of ancient biology, for the first time sampled to try to find useful things in it. And we did a lot of in vitro testing, and this is the preclinical mouse data testing. Again, the abscess infection model here, the deep thigh infection model here. The top candidates came from the woolly mammoth, a molecule that we're calling mammutocin 2, and also from the ancient sea cow, the giant sloth, and the extinct giant elk. And in some cases, some of the best molecules that we found, they decrease infections to the level of polymyxin B, which is a last resort antibiotic that we have available in the clinic. So that was exciting to see that level of efficacy as well. This made the cover of the journal, where they highlighted this new concept of really trying to look at extinct biology for new solutions for antimicrobial resistance. Okay, so why molecular de-extinction as a framework? So I'm just going to tell you briefly things that we've learned so far. It has enabled us to explore a completely new sequence space of molecules. So if you do a multi-dimensional visualization of every compound that we found by sampling ancient biology, and you try to overlap it with living biology today, a lot of the things that we found are outside of the boundaries of living biology, meaning we have uncovered new sequence space of molecules, therefore increasing the molecular diversity that we thought was possible. We think that a lot of these peptides that we're finding, they play a role in immunity throughout evolution, and we're currently testing that hypothesis. And finally, we believe that bringing back molecules from the past can help us address present-day problems. So as you can imagine, contemporary pathogens that kill millions of people today, they've never seen molecules that existed thousands or hundreds of thousands of years ago. So we're literally bringing back a whole new arsenal of potential therapeutics to counter present-day infections. And again, you can apply the same paradigm for bringing back a whole new arsenal of anti-cancer therapeutics, or things that might help with neurodegeneration, or things that might help boost immunity, and so on. A couple of ramifications from this work. On the bioethical side, when we were initially uncovering all these compounds, as you can imagine, it was really exciting scientifically, right? Like, we're doing this for the first time. But then I started worrying a little bit when we found that a lot of the molecules some of the molecules that we're finding, when we do multiple sequence homology with living biology, we couldn't find them. Meaning a lot of these compounds that we've identified in ancient biology, they're not expressed in the living world today. So we're literally resurrecting some of these things using chemistry in the lab, right? Is it okay for us to do that, right? From a bioethical standpoint. So we've been consulting with bioethicists to make sure that we innovate, but that we do so responsibly. So one of the interventions that we've incorporated into our workflow is that if we identify something that might look like a bioweapon or like a biotoxin, we just don't synthesize that. We don't pursue that further. That's one of the sort of interventions. And then on the patenting side, when we were initially uncovering all these molecules, I went to PCI, the patent office at Penn, and I told them that I was aware that natural sequences are not patentable because they belong to humanity. But then I asked them, what about all these compounds that we're finding that used to exist in the world but are no longer expressed in biology today? And this is actually, it's opening up a whole new area of patent law, because patent lawyers don't know whether we should patent some of these compounds. So it's quite interesting to see how that field is evolving as well. In the meantime, I've written with a colleague this piece in Nature Biotech on some of these musings. In terms of next steps, like I mentioned, we conceptualize biology as just a bunch of information. So we want to mine the world's biological information to try to find new antibiotics, and then move to in the future, to other useful molecules for cancer, immunology, neuroscience, and so on. And so we want to sample the whole tree of life. Initially, we focus on eukaryotes in the form of modern and ancient humans. So next, we wonder whether we could explore all of the bacteria in the world, another huge branch of the tree of life, right? So I'm going to briefly tell you about two collaborations. This is one of them where we use machine learning to explore virtually, a huge component of the microbial life on Earth, almost 90,000 microbial genomes and over 60,000 metagenomes that come from many, many parts of our planet. And using this ML model, we're able to discover almost 1 million new antibiotics by sampling through this microbial dark matter. Remember, it's usually very hard to find just one or two. And here on the computer, we're able to find almost 1 million, which was really an amazing moment. We've released this publicly to everyone through this website that we call NMP Sphere. So every single sequence that we've discovered is available for everyone to look at. And we did that with the hope of, amongst all of us, also all the scientists in the world, hopefully amongst all of us, we can transition some of these molecules into the next stages of development. And in my lab, we're just around 20 people, 20 plus people. We can only do so much. Imagine amongst all of us, millions of people around the world, what we can do. So that was the concept there. We found preclinical candidates. Here are two examples. One comes from enterococci that were effective at reducing infections in mice. And this is the other collaboration with our colleagues at Stanford, where we decided to look at human microbiomes as a source of new antibiotics. We thought that human microbiomes would be an ideal place to look for this because these are like good bacteria that coexist among many other good bacteria, but we're talking about billions of them, right? So as you can imagine, if you're part of one species, you need to take over a particular niche, and then you need to replicate and grow, right, into little microbial towns, and then microbial cities and countries. So you need to take over a particular niche, and that means you need to outcompete other microbes in your surroundings. So we hypothesized that one way that microbes did this is by producing small peptides that enable them to fend off surrounding microbes in their vicinity, sort of like a type of microbial warfare, like chemical warfare. And that turned out to be correct. So we found that individual microbes produce individual little weapons that they use to sort of shape and reconfigure the environment around them. And again, we found preclinical candidates that were effective at reducing infections in mice. Prevotellin-2 is a molecule that we found produced by the gut microbe Prevotella copri. That, by the way, is a microbe that we all have in our intestines. So this is another reminder of the amazing capabilities of microbes, right, to invent and come up with new chemical tricks. And finally, this is still a preprint, but we sample archaea, the final branch of the tree of life that we hadn't done completely digitally. We did it with Apex, with the AI model that I described. And we uncovered a new class of antibiotics that we call archaeocins. Again, this is important because typically antibiotics have been found in bacteria and in fungi, but archaea have never been really systematically explored as a source of really anything, let alone antibiotics. And the really amazing thing is that we got a 93% hit rate based on this is experimental hit rate based on the prediction made by the model. My current estimates is that if we get to 95+% hit rate of accuracy, I think we will barely need to do any experiments. I think we'll reach a point where we can rely on the model sufficiently to really diminish the number of experiments that we have to do to validate things. And that's, I think, in the future, hopefully we go more towards that in that direction where we can use these tools really reliably, right? Okay, I'm going to just mention a couple other recent preprints that I'm really excited about. One of them is Apex Go. So here we use Apex, our AI model, but on top of it, we build generative AI system. And this is what we did practically. So we can start with a template here, this red cross that we give the machine. And the machine then the task that we give the machine is to improve that template, to make it a much better antibiotic. So it's lowering the MIC, the predicted MIC, the predicted antibiotic activity. So the lower the number here, the better. So we start with this template, we give it to the computer, and then the computer samples through sequence space, and it produces all these synthetic variants. All these little clouds are completely made up by the computer. And the computer predicts that all of these are much better than that, right? The lower you go, the better, right? Just, on the computational side, this is the first experimental validation of generative Bayesian optimization in any setting, which was really exciting from a CS perspective. And we got 85% hit rate and 72% success rate and enhancing the antimicrobial activity. So 72% of the molecules made up by the machine were better than the template. So this is a way of like doing optimization fully in silico. And of course, we're doing the experiments just to validate the computer's assumptions and the predictability. But imagine if you could take an antibiotic and improve it completely in silico in a few hours. Imagine if you can take an anti-cancer drug and improve it in a few hours, or any kind of molecule, right? This is the promise that this has. We're all familiar with multimodal models, right? Like ChatGPT, you can do comprehend text, you can do text to image generation, you can do text to video generation, some of these models. So we're all very, this is pervasive in our society, but people have not really applied multimodality to molecules. So we wanted to do that. So this is a preprint still, but we developed this model, Apex Duo, where the whole goal was to design multimodal molecules, and the proof of principle scenario that we wanted to address was intracellular infections. These are bacterial infections that bacteria that enter our cells, our human cells, and they replicate inside. And so they infect us. But then our immune system doesn't work very well at resolving them, right? Because they're hiding inside our cells. And then conventional antibiotics can't really enter our cells very efficiently and clear the infection. So we wanted to design peptides that could do two things: penetrate into our human cells, and then target the bad bacteria that are inside, replicating. And that's what we did. We, through this encoder-decoder model, we're able to incorporate and integrate these two different functions and create new molecules that are completely made up by the computer. And a couple of them, the hit rate was pretty low, but 3 of them out of, I think, 30 that we synthesized, 3 of them worked, and 1 of them worked quite well. So that one we characterize in detail in this paper. We fluorophores, we tagged it, we followed how it penetrated into our cells, we followed how it killed the bacteria. So admittedly, the hit rate is still quite low in this, but as you can also imagine, this is a hugely challenging problem, right? We're trying to incorporate 2 functions in a single molecule for the very first time. And so I think this is just the beginning of, I can foresee in the future having medicines that have more than 1 function. So you can imagine having an anti-cancer drug that also reduces inflammation or something like that, right? So just to sort of wrap up, right? Traditional methods of antibiotic discovery, you can take up to 6 years to come up with some candidates. That's a very sort of unreliable process. Today in my lab, in a few hours, we can discover hundreds of thousands of candidates, and we can go from discovery to synthesis and testing within a week if we really push our resources. So on any given day, like today, I came in the morning, I had a cup of coffee. By lunchtime, my team told me we had like a bunch of new molecules to play around with. And now by dinner time, I'm going to have a ton of other molecules to play around with. So it's really like a playground for a scientist like myself. I've been dreaming about this for my whole life. And now it's really fun to be able to finally have this technology sort of at this stage. And this is sort of the impact of the work so far. We have accelerated by 3.5 million times our ability to discover antibiotics. It's been estimated that the time saved with this research, it's about over 1 million years of research, and the equivalent of 100,000 PhD students working for 6 years each. So this is what we can do today with AI in the field of antibiotics. It's really exciting, and I can't wait to see what we can do in the next 5 years or so. It's a very young field, so looking at publications over time, until 2018, there was barely any papers, and now this is growing sort of exponentially now. So it's also a very young field, but hugely exciting, and It accepts and welcomes people from many, many different disciplines. Like in my lab now, I have microbiologists, but also computer scientists, chemists, engineers, all working together. But this is sort of the summary of what we've done. I didn't have time to cover all of this, but this is sort of a lot of the advances in antibiotics AI that we've been able to achieve. And these are the preclinical candidates that we've validated for safety and efficacy so far. So these are at the preclinical stage. And this list continues to grow every month or so. So the dream that we have is that hopefully we see some of these transition to clinical trials in the future. The other thing that I'll say, and maybe you've perceived that from my talk, but I believe that this is the coming of age of peptide design. Peptides are the simplest, smallest functional molecules that exist in the world. And we've shown that if you build good, robust data sets, you can build AI models that are like incredibly powerful. And I think that's sort of the future. And I foresee a world where peptide design can be applied not only to antibiotics, but let's see here, but also to many other fields. We're already working in immunology. We have a project in neuroscience. We have a cool food science project where a brewing company, I cannot say the name, but they basically, we have this collaboration where they ask us to apply one of our AI models to find anti-inflammatory peptides in grains that they use during the fermentation process in ways that we can predict where they get cleaved off by proteases and they get released through the fermentation process, with the idea of making an anti-inflammatory beer that is also non-alcoholic. So imagine, like, for people that have issues with inflammation, when you drink beers, you might imagine having an anti-inflammatory beer. So that is it's close to what they call clinical trials, which is they get people to drink one beer a day for a month, and then they do like blood work to see whether the inflammatory mediators go down throughout that period of time. Those are clinical trials in the brewing industry. I don't know. We're working on agriculture as well for crop, designing peptides against pathogens that affect crops, mastitis infections in cows. We have a couple of anti-cancer projects. And we've done some work on diagnostics, drug delivery with Apex Duo, but we haven't done work in vaccines, semiconductors, or tissue engineering yet, but those would be clear application areas as well. And with that, I'd like to thank lab members, past and present, our great collaborators from all around the world, our funders, and then I'll leave you with links to our website, email, and social media. We'd love to get in touch by any of those means. Always love talking science. And again, thank you for having me, and would love to take questions.
Definitely have time for questions. I do have a microphone to questions we can do that are folks online. I have two back here. Turn them over.
Okay, that was a great talk. Thank you.
Thank you.
My question is either in your AI models or in your sort of validation testing, have you accounted for antimicrobial resistant organisms, or are you using sort of the susceptible bacteria, since the target would presumably be those like multi-drug resistant organisms?
Yeah, great question. A lot of the in vitro testing that we've done and validation experimentally, we've done it with multi-drug resistant clinical isolates, or at least intrinsically resistant, like Pseudomonas, for example, right? That we know are intrinsically resistant to a number of antibiotics. Resistance development assays to our peptides. We've done that with the human proteome work. We tested 3 of the encrypted peptides So we do this resistance evolution experiment where we expose bacteria like Ewamani or Pseudomonas to sublethal levels of each of our compounds, and also antibiotic controls like ciprofloxacin. And we track evolution throughout those 30 days. And in those experiments, we saw the bacteria evolved resistance to ciprofloxacin, which is a fluoroquinolone, in like 2 or 3 days, which was insane to see in the lab. I mean, ciprofloxacin is used everywhere, right, in the hospitals. But we didn't see resistance evolution to our peptides throughout that period of 30 days. That doesn't mean that we wouldn't see it in the future, but that's promising data. We're now, I have a postdoc in the lab running a huge resistance experiment where we're testing many peptides and looking at their resistance profiles. We're using a hypermutant strain of E. coli, a mutant for that. We are forcing evolution to see actually resistance pathways accelerated so that we can actually try to predict that in the future. The goal of the project is twofold. We want to, first of all, build a data set to then have different resistance evolution trajectories and profiles to train a AI model that can be used to design resistance-proof molecules, or as resistance-proof as possible. And the other side of that project is we're going to be sequencing all the genomes of the mutants to see what what mutational landscapes take place. So yeah, we're working on all those areas.
Cool, thank you.
Thank you.
Great talk.
Thank you.
I was also curious about resistance, so thank you for asking that. I'm also generally curious about mechanism of these antimicrobial peptides. Like, are they directly lytic the way that, mucosal antimicrobial peptides are? And you think that's why you're not getting the sort of like enzymatic based resistances that you get with other antibiotics? Or do you have other mechanisms that you found for your different targets?
Yeah, good question. We're a bit limited by the mechanism of action assays that we have in my lab right now. We have some fluorescent assays and biophysical assays where we look at membrane targeting, mostly outer membrane, like permeabilization, cytoplasmic membrane depolarization in gram-negatives. So we are limited by what we can do. So everything that we look at is membrane targeting. We're starting a collaboration with colleagues over in Boston where we're going to do transposon sequencing to really do a thorough genomic study on things, plus the experiments that we're currently doing where we're going to sequence the genomes of mutants to try to understand a little bit better. But admittedly, so far we look at membrane targeting. So depending on the sequence, the mechanism varies a lot. Some of them are able to target both the outer membrane, the cytoplasmic membrane, some of them just one or the other, or some of them none. And we don't really know how they kill bacteria at this point. But
Okay.
again, we've synthesized thousands of peptides so far. We've tested them in vivo. We've tested hundreds by now. So we've done a ton of work. It's just we're a bit limited on the mechanistic side.
Thanks. Based on that, do you have, for the peptides you've come up with that work on intracellular bacteria, is there any mitochondrial toxicity that you see, right? Mitochondria or just endosymbionts?
We haven't done, so one project that we have done, we've used Apex to sample mitochondrial genomes to see if we could find antibiotics encoded in those genomes. And we have data for that, but we haven't tested off-target effects on mitochondria in experiments, but we have found antibiotics encoded in the genomes of mitochondria, which I think is kind of interesting if you think about it, right? Mitochondrial ancestral bacteria. And so we're trying to think about that, how to explain that, but that's all we've done with mitochondria, so far. I think they're amazing. They're like really cool organelles.
Yeah, we have a couple questions online real quick, and then we'll jump over here. Sure, if you want to unmute and see if you could ask. You're muted. You're still muted.
Yeah. This is a great talk.
Looks like we're not going to hear you. Can you type?
Can you hear me?
And then I'll ask them for you.
Can you hear me?
Meanwhile, we'll go over here.
I have 3 questions. I may have missed it. What are your criteria or selection criteria to call it an antimicrobial peptide? Is it based on known antimicrobial peptides, or what's you never mentioned really how selection criteria?
So I didn't mention it because it depends on the algorithm, and you have to get into all the details for each of the algorithms. So the talk would take like 3 hours. But so for example, for the scoring function that we used to sample the human proteome, we took into account, we wanted to see what was the minimal sequence that will interact with a bacterial membrane. In order to do that, we use this equation, taking into account two physicochemical parameters, one of which was Heisenberg's hydrophobic moment, the other one, which is the alpha helical propensity based on Pascal scale. So in English, we wanted to, it was a particular ratio between positive charges and hydrophobicity, because we know from many experiments and chemical knowledge that if you have that, that minimal peptide will interact with the membrane, with a bacterial membrane, which is negatively charged, typically on the surface. And so first through electrostatic interactions between the positive charge and the negative, and then the hydrophobicity will allow it to translocate into the membrane. So that's what we did for that initial work that we published in 2022. But then Apex was trained on in-house experimental data. So basically Apex sees all the sequences of amino acids and sees all their antibiotic activities against many different microbes. And it learns what are the patterns that I need to, what are the patterns, hidden patterns that make functional molecules that are antibiotic molecules. And so that's what it does. So it's a deep learning model in that case. And then what we've done subsequently, we've built thresholds, filters that you can apply to the algorithm, where you can say, if this looks 75% similar to something that is already known, discard those sequences and just give me something that is novel. So you can create novel, you can sort of bias your algorithm to find novel things by using those filters. Or with Apex Go, it's a generative AI model, so you're creating entirely new sequences that have not existed in nature, that do not exist in nature. So those are some of the. It's just like every model is a bit different, so it's a little hard to explain all of them in a way that is like reasonable. But yeah, those are some of the things that we've done. Yeah.
You exclude possible toxic for humans.
Yeah, great question.
Or you are still then stuck with it. Okay, now we have.
Yeah.
It's not toxic for humans.
Yeah, great question. We don't have enough data to build an AI model for cytotoxicity or hemolytic activity against red blood cells yet. We are building those data sets at the moment. And my goal in the future is to build a super algorithm that can optimize for antibiotic activity, but also counter select for toxicity, optimize for stability. So we also have we're building data sets for stability, where we expose the peptides to human serum and we track the profiles of degradation over time. And we see, depending on the sequence, the degradation profiles. And so we are building all these different data sets in parallel in different projects. And the ultimate goal is to then do it like we did with Apex, right? It took us 3.5 years of very hard work to build a data set that finally was good, to train a model. And we're trying to do use that same paradigm for all these other things, like toxicity, stability, PK/PD profiles, all of those things.
My final question is, testing, bacteria is so different. Your hit rate for intracellular levels is very low. How do you exclude, are you testing it with 3 bacteria? If it's positive or negative, but it may be positive with the fourth one. How is the selection criteria?
For that particular paper, it actually took a long time. So there was a lot of groundwork in the Apex Duo paper, because we had to first look at what bacteria to target. So we looked at our model and its accuracy against each of the intracellular pathogens like Salmonella, Staph aureus, E. coli, or some of the ones that could be intracellular. The best accuracy metrics were against Staph aureus, so that's why we selected Staph aureus for that particular project. Then we had to develop this Transwell experimental assay to look at penetration into mammalian cells, which was non-trivial. That took multiple months to develop the assay to then be able to test experimentally penetration into mammalian cells of our peptides. So in that case, that's what we did. So Apex is a multitask learning model. I didn't mention that. That means that it has the capability of predicting, if you give it an amino acid sequence, it has the capability of predicting antibiotic activity against dozens, I think it's like 16 different pathogens. It's not great at all of them because our data set is still a bit unbalanced, meaning we don't have as much data for gram positives. So it tends to be worse at predicting activity against gram positives, for example. But those are problems that will be solved, I predict, by just improving the data sets as time goes on, right? But Apex is multitask learning. And so in theory, you can give it like amino acid sequence, and you can ask it what is the likelihood of that being active against E. coli or Pseudomonas, and it will tell you. It will tell you a number. Then you still need to do the experiment. We're still not at that 95% plus accuracy that I think is needed to really fully trust the models. But yeah. Yeah.
One thing we discussed, toxicity. There's another issue. When you could produce a molecule which has anti-insulin
Mm-hmm.
or something which is not toxic, but which a lot of peptides used in different biochemical and physiological things in the body. So you might suddenly, do something. Is there a way to try to, there must be a way.
Mm-hmm.
I eliminate everything which is known in terms of peptides.
Yeah. It's a good question. I mean, how can we eliminate every single off-target effect? I mean, it's a tough question, but one thing we haven't done yet, for example, is we haven't done the experiment of whether antibodies get generated when we expose human cells to to peptides. That's something that we should do at some point. It's on my to-do list of things that I know we need to do, but we haven't had the time to. That is another good one. Like, what are all the off-target effects, potential off-target effects, and how can we counter-select against them? We're still not there. But if there were public databases with each of, we could also use public data. Like, we try not to use too much public data because it comes from many different labs done differently, and so in our experience, when we're trying to build models with just public data, they're not as good as when we generate all the data. But of course, it's way faster because it's a multi-year project every time we have to generate all the data. It's a lot of work. So yeah, hopefully in the future. Yeah.
Maybe missing something. How do you generate proteomics data from extinct organisms? Yeah, very good question. So this is a field that has been evolving over decades. As usual, we stand in the shoulders of giants. So there are many people that have been working on sequencing methods for archaic DNA. Perhaps the pinnacle of that field was 3 years ago when Svante Pääbo got the Nobel Prize for developing sequencing methods for ancient DNA. So people, over time, they've been devising all these sequencing approaches to basically try to amplify, archaic DNA, as you can imagine, is very degraded, right? And so initially people developed methods to amplify mitochondrial DNA because we have multiple copies of mitochondria. So we have generally a lot more genetic material than if you're trying to amplify chromosomes. And later on, the chromosomal amplification improved. But basically, the good news for us is that a lot of those data are available in databases, and we can, it's just code, right? So with the right algorithms, we can just sample the code.
I think we need to stop there for our space reservation, but if you were online and were not able to ask your question, we apologize.
There are like 19.
You connect Cesar on the social media that he's posted there, or just send me an email, or you send me an email and play it on check. But let's thank our speaker.
Thank you.
If you need a parking ticket, from Bradley,